Okay, so let's get back into it. Um, what if you don't have enough glucose to make ATP, right? You are you just like, okay, well, I can't make energy, I can't make ATP, I'm just gonna die. No. So you have to have a backup method, right? So we went over carbohydrate metabolism. You should take the time and go through that. But let's cover non-carbohydrate precursors. So what happens when we run out of carbs? What is our second fuel source? Good, it's going to be lipids. And when we run out of lipids, what is our third fuel source? It's going to be proteins. Okay, so you see glycerol and fatty acids and then amino acids. So let's take a look. So in order to use fats, we have to break up the triglyceride. If you remember back to triglycerides, it's glycerol plus 3 fatty acids tails. So you have a glycerol backbone and 3 fatty acid tails. The glycerol is going to get put into glycolysis and used and just plug and chug. The fatty acid tails are going to get ripped off into carbon segments, right? 2 carbon segments, and stuck in as acetyl-CoA. Proteins are a different story. You break down the proteins to amino acids first, and then before you can use that amino acid, you have to rip off the amine group, right? That amine group has to go bye-bye because you can't shove a nitrogen-containing group into metabolism. It just doesn't work. And so let's look at those processes in a little bit more detail. So triglycerides are the most common energy storage molecule and the second most common used fuel for cells, right? So after a meal, whatever fatty acids you have or whatever sugar you have gets converted to fatty acids and they're stored in the adipocytes. When you need them for fuel, triglycerides break down into glycerol and free fatty acids. We call this beta oxidation. So it has a specific name. Excuse me, it's called beta oxidation. Um, those free fatty acids are then dumped into the bloodstream, and your skeletal muscle can use them. Your liver, this actually occurs, this beta oxidation occurs in the liver, so they can use them. And we'll talk a little bit about this in more detail. Triglycerides, so fats, they yield twice as much energy than carbs or proteins do, right? So um, some of you have done a nutrition assignment, and when you do the nutrition assignment, you know that carbs and protein, for every gram of carbs and protein, you make 4 calories, right? Triglycerides are different. For every gram of triglyceride, you make 9 calories. Okay. So, because you make more calories for fats, you need to eat less fats in your diet, typically speaking. Okay, here's the pathway. So if you go up to the top right side, adipose triglycerides are broken down and released as free fatty acids. They're either taken up by cells and broken down by a beta oxidation into acetyl-CoA. Um, they're either enter citric acid cycle or they're converted to ketone bodies. Okay, so if you look at the picture, here's your triglyceride. The glycerol gets broken down and thrown into a glycolysis. We call that gluconeogenesis, making new glucose from a precursor. And then those fatty acid tails get broken down into 2 carbon segments and thrown in as acetyl-CoA. Those free fatty acid tails can be, let's say, 20, 22 carbons long. And so if you imagine if you're taking a 20 carbon fatty acid tail and you're chunking it into 2 carbons, you're going to have 10 acetyl-CoAs. So that's going to be 10 acetyl-CoAs making all of the ATP from the electron transport chain. And so that's a lot, right? This process name is called beta oxidation, right? You can also build fats up in the reverse direction. So if you need to make fats because you have eaten carbs or proteins in excess, we call this lipogenesis. But what I want to focus on right now is that acetyl-CoA. So acetyl-CoA, beta oxidation occurs in the liver because the liver has enzymes to do this. Acetyl-CoA will go through Krebs citric acid cycle um, for the liver and make ATP for the liver. If you do not have enough carbohydrate to keep using that for fuel, your body will use fats. How do they use fats? Your liver is going to break down the fats to make acetyl-CoA, and then your liver is going to convert them to ketone bodies. We call this ketogenesis or ketosis, right? So you're going to take acetyl-CoA and make ketone bodies. The liver does this. The liver is going to take those ketone bodies and dump them into the blood so that your body can take them up, or so that your cells can take them up. Your brain prefers to use glucose, and secondary, it prefers to use ketones. And so your brain is going to scoop up those ketones and use them for fuel. So when do we use fats for metabolism? When we've exhausted our carb sources. When we've used the glucose in our bloodstream, when we've used the glucose that was stored as glycogen in the liver. Usually you have enough glucose stored as glycogen to fuel about 8 hours of not eating, right? And then you start to break down fats. And so how can we tell that we're breaking down fats? You could actually urinate on a urine dipstick and it would test positive for ketone bodies. And so you can measure if you're excreting out ketone bodies, and that would be an indication if you're going through ketosis and using fats in your body versus using carbs. This is okay at a low level, right? So let's say you skip breakfast and lunch. You are now burning fats and you're making ketones for your brain to take. The problem though with ketones, and this is important, is that they are acidic. And so low levels of acidity, your blood can buffer, right? Your blood has bicarbonate, your blood has those negative albumins that will help buffer the blood. But if you make this an excess, let's say with somebody who has diabetes mellitus, and they can't get sugar into their cells, they can't get glucose into their cells, so their liver is making excessive amounts of ketones that can damage organs and acidify the blood. So ketones are okay at a low level, but you have to remember that they're acidic. And when you're dealing with pathological conditions like diabetes mellitus, you're going to make excess ketones, and that's going to acidify the blood, right? Let's look at protein. So after a meal, proteins are broken down into amino acids. They're pulled into the cells in the liver. Your amino acids are used to synthesize whatever you need in the cell. And so they're going to be used last as a fuel source. They're not stored in the body as amino acids. They're converted to glycogen or triglycerides for storage. Let's talk about how you actually use these to make NAD plus. Okay, so either you can stick those amino acids into intermediates of Krebs, and they can plug and chug that way to make ATP in the liver, or they can go through oxidative deamination to make NADH and ketones. So here is using the amino acids, just throwing them in at selected spots to go through Krebs and citric acid cycle, right? That's one way to use amino acids, and that's one of the main ways. The other way is oxidative deamination. You can't have an amine group or that NH2 on the amino acid. So you're going to pull it off of glutamic acid, and then it exists as ammonia when you pull it off, and then you're going to convert that to urea, right? So let's take a look at the picture. I know this is a little busy, but here's transamination. So trans means you're pulling the amine group. So transamination means you're pulling the amine group from one amino acid to another amino acid. So look at how we stuck it on this alpha ketogluteric acid and we made glutamic acid. Now that we have the glutamic acid, we're going to go through oxidative deamination. You're going to use NAD plus to make NADH. In the process, you're going to rip off that NH2, and it ultimately is going to get converted to NH3. So basically, it's a process of where you're ripping off the NH3 or excuse me, NH2 to get the amino acid by itself, um, so that you can use this for fuel and so you can regenerate, so that you have NADH. Ammonia, let me say something about ammonia. So ammonia is a byproduct of protein metabolism. You can't just leave it in the blood because it will cross the blood-brain barrier and alter your mind. So it is a toxin. Ammonia in your liver gets converted to urea ultimately. So one negative byproduct of protein metabolism is ammonia production and making urea. This happens in the liver. If the liver is damaged, when you're trying to break this down, sometimes you can't convert the ammonia to urea, so you get ammonia poisoning. Okay, so starvation, what ends up happening? So you need to be aware of starvation for adolescent, right? Women or adolescent girls, usually prepubescent, because their body's starting to change and they often, well, they starve themselves, as well as geriatric patients. So whether they're shut in and they're not getting enough food or nutrition, or they have a GI tube, a feeding tube, and they're not getting enough nutrition that way, enough protein for healing. All right, so this consists of 3 phases. So the first phase, the blood glucose is maintained by breaking down glycogen. The glycogen is stored in the liver, your body will break it down, and you have enough glycogen to maintain blood sugar probably for about 8 hours, 8 to 12 hours. Once you've exhausted that, what happens? You're going to use lipids primarily and a little bit of protein. So you're going to start breaking down fats in the body, um, through beta oxidation to make ketones. Um, and then you're going to break down proteins after that. Proteins, the amino acids, you have to rip off the amine group to make ammonia, and then you make urea from that. The brain doesn't store glucose, so it freaks out or goes into shock when it doesn't have enough glucose or enough ketone fuels. Your second phase is fats are primary energy source, so your liver is going to convert fatty acids to ketones. We call that ketosis. The ketone bodies, you can always tell when somebody is going through ketosis, when they're starving themselves or not getting enough nutrition, because they have this distinctive smelling breath. And so when you get close to them, you can actually smell their breath, and it'll smell like fermenting or alcoholic bananas because of those ketone bodies. The ketone bodies are kicked out of your respiratory tract, of your respiratory lining. The brain will use the ketone bodies as long as it can, and this can last for several weeks. The third phase is using proteins. So then proteins are used when glucose is depleted and your fats are depleted, right? So your body is going to start to degrade the cellular proteins and your muscle to make NADH and ketones. At this point, people usually die from a secondary infection as a result of this long-term starvation. So it's not uncommon when somebody is starving or not getting enough nutrition that they die because of pneumonia because their body wasn't healthy enough. The last thing is diabetic ketoacidosis. So if you are diabetic, glucose can't get into the cell because you have a lack of insulin, right? Excuse me. So your body cells are going to use lipids and proteins as a fuel source. They're going to make ketones, right? So what ends up happening is because you can't get glucose into your body cells are screaming for fuel. And so the liver upregulates breakdown of fats to make ketones. And so this happens on a massive scale. And so you have ketones getting dumped out, ketones getting dumped into the blood, ketones getting dumped into the blood. Eventually, if you look at the last bullet, it's going to acidify the blood. Okay, so what happens when the blood becomes acidic? Or actually, what is the pH of blood? Good. So the pH of blood is 7.35 to 7.45. What happens if you have all these acidic ketones accumulating in the blood? What happens to pH? Does it stay at 7.35 or does it go higher or lower? Good, it starts going lower. So 3, 7.25, 7.2, 7.15, you get down to 7.2. And what happens? Does hemoglobin want to bind oxygen, or does it have a really hard time binding oxygen? So as you drop the pH, as you acidify the blood due to the ketones, hemoglobin has a really hard time holding onto oxygen. And so you're messing with the oxygen availability of those cells. Okay, so what are you going to do? I want you to make sure that you can draw out everything, right? Make sure that you can draw out Krebs, glycolysis, electron transport chain, and make sure you know how many ATP are made at each step. And then go over uh, um, fat metabolism and protein metabolism. And make sure that you have a quick condensed version so that when you get an exam, you can take a scratch piece of paper and draw it out really quickly so you can answer your exam questions. Okay, thank you for your time. This ends our recording.